I love Atlantic salmon because of their resiliency and also how they connect our freshwater and ocean ecosystems.
As a kid, I started trout fishing, and of course, the salmon are bigger, they're shinier, they're flashier. It was the big prize.
They're such a beloved species by so many people. I'm not an angler myself, so I don't angle for salmon, but just seeing so many people who are so passionate about the fish, and I can understand why. They're just a really fascinating and beautiful fish.
My name is Marian Stubbings. I'm a Resource Management Officer with Cape Breton Highlands National Park and the coordinator for the Atlantic Salmon Recovery Project. Atlantic salmon have long been a species of significance in Eastern Canada, especially to local communities and Indigenous people who often relied on them for traditional practices and as a food source. Atlantic salmon were able to support a commercial fishery in the past. Off the coast of Cape Breton, they used to do fishing using gill nets. However, this practice ended in the 1980s as part of the effort to conserve the species. Nowadays, the fishery continues, but it's more of a recreational industry done by fly fishermen on scheduled rivers. The Atlantic salmon that live in the Clyburn Brook are part of the Eastern Cape Breton designatable unit of salmon, which is a distinct population that has been listed as endangered by Acoustic, which is the Committee on the Status of Endangered Wildlife in Canada. Atlantic salmon have been declining across the range for the past few decades. We have an ecological monitoring program that tracks the return of adult salmon coming back to our brook every year. And we also have started a program with our Salmon Recovery Project that is looking into determining the population numbers of smolt, which are the juvenile fish leaving in the spring. And that's where we're seeing that the smolt leaving, it's way fewer adults that are returning from the ocean. So for us, the main threat is the changes that are happening out in the ocean, which right now are not completely understood, but are having an impact on our salmon. We have been monitoring the Clyburn Brook population specifically. We've been doing these snorkel surveys where we track adult returns since the 1980s. We'll call them good salmon returns. So this is when salmon returning were meeting a conservation requirement. That's a requirement set out by the Department of Fisheries and Oceans. For our river in particular, it's 75 salmon. However, since the 1990s, we've seen a 95% decrease in the number of salmon returning. So that means in 2018, we only saw 8 salmon. In 2019, we only saw 5. On the Clyburn Brook, we are undergoing Smolt-to-Adult Supplementation as one of the main activities in our recovery project. So Smolt-to-Adult Supplementation means that we're capturing wild smolt as they're making their migration out to sea, intercepting them, and we are partnering with the Dalhousie Aquatron, where they will be raised in captivity until they reach maturity. At this point, they'll be re-released into the Clyburn Brook just in time to participate in spawning in the fall, with the goal that they will help spawn the next generation of wild fish.
My name is John Batt, and I'm the manager of the Aquatron Laboratory at Dalhousie University. When the Salmon Conservation Project started, it's a national project with 5 national parks in Eastern Canada. One of those parks is the Highlands National Park, and each park had to have a partner that can hold fish. And we were visited by Parks Canada a few months before the project started. They were looking for a group that they could bring the fish to, and we could rear them until they were ready to get them back. And we were chosen to be the center that does that. The Aquatron is probably the largest aquatics research facility in Canada. We have over 75,000 square feet of research space for people wanting to work with water and fish and all the things that go with that. There's so many great research facilities around the world and in Canada and regionally, but What makes us unique is the ability to work at almost any level. So we have the really big tanks, 50 feet diameters, and then we have all the labs where we can do really controlled experiments at a small level. So we can sort of do just about anything in between too. So that plus our staff is really special because we've been doing this a long time. So it's a long legacy of people passing information on to people. So we have this just huge full-time staff that's really experienced, really professional, and really good at what they do. The parks people have been around visiting different centers. They came here, they really liked what we do. They were able to see other projects that we do that are similar, so they were pretty happy with that. So then they put out a contract for us, and we took that on. And we started bringing in the first fish fairly soon thereafter. The first return was done a couple years later, but over time we built this relationship with the parks people. We go up in October, it's so just exciting to go there. We bring a truckload of fish that they haven't seen in a year and a half. They're big fish. It's a beautiful time of year to be in Cape Breton, so it doesn't take long to build great camaraderie with those people, great relationships. And then we started talking about what else can we do for the project? What could Del bring to the table? And we brought in Dr. Robert Lennox.
My name's Robert Lennox. I'm a professor at Dalhousie University and scientific director at Ocean Tracking Network. At our core, OTN is a network, and we collaborate really closely with lots of different organizations, from academics to government to non-governmental organizations. So we loan infrastructure, we have our data system that pulls in data from all sorts of different partners, and we aim to really facilitate getting the capacity to track animals out into different investigators' hands. So this is an acoustic receiver, and OTN maintains lines of acoustic receivers around Atlantic Canada and in British Columbia, and we also loan out thousands of these receivers to researchers around the world. Because we can't use GPS to track animals under the water the way that researchers can use to track birds or animals on land, we have to use sound. So the sound that's transmitted by a tag gets detected and logged on a receiver like this. We use electronic tags that have a battery, and the battery powers the transmitter that actually sends out the signal. So we'll implant a transmitter. That transmitter will send out a signal about every 2 minutes as the animal swims around. And when it passes one of these receiver stations that we've deployed with one of our partners, it'll log the ID of that animal that the tag is sending to it. But once every year, we'll download all of the receivers, we'll pull out all of those into our database at OTN, and we'll be able to figure out where each of those animals has moved around that network of receivers. The tags that we use are relatively small, but we match the size of the tag to the animal that we're actually tagging. I remember when I first started at Dalhousie, John Batt and I started to talk about this amazing project that he had started with Parks Canada to help rear these Clyburn Brook Atlantic salmon. I remember very distinctly John telling me that he thought it would be so validating and so valuable to know if those fish were remaining around where they were released, if they were moving upriver even to find spawning grounds, kind of continuing a migration. It would really demonstrate that they were exhibiting some natural behavior. And so John and I started to think about how we could actually address this. And fortunately, Sarah Penny and Maren Stubbings from Parks Canada, they were super enthusiastic about these questions because they've been supporting this project, and they also wanted to know how is this functioning? Has this been an effective strategy for the conservation of Clyburn Brook? And so we were able to bring on Natalie Koopman as a master's student and to implement this project that we had kind of envisioned.
My name is Natalie Koopman, and I'm a master's student in biology here at Dalhousie University. I applied for my master's with Dr. Glenn Crossan and Robert Lennox. And Parks Canada really came into the picture as they were already partnered with the Aquatron. This is where the project came in, where we were really hoping to see how these Atlantic salmon were behaving, if they're migrating the same as their wild counterparts. And this is where the project really blossomed and the partnerships began. And past years, when we see Atlantic salmon decline like this, supplementation programs are started where Atlantic salmon are grown from eggs till they're juveniles and then released back into the river. But this doesn't bypass the marine environment, so they're still subjected to that bottleneck of the ocean where we're seeing very few return from. And it's also known to have many genetic and behavioral consequences as well. So when Atlantic salmon are raised as juveniles in captivity, they might not know how to feed the same, they might be more susceptible to predation, they might not know the exact same migration route. It can be quite problematic and potentially have the exact opposite effect of what we're looking for. So instead of turning to traditional supplementation programs, Parks Canada decided to test SAS, or Smolt-to-Adult Supplementation. And instead of releasing the fish as juveniles, we allow them to grow up naturally within their wild river with all of their natural instincts. They get the scent of their river as well. We then capture them, raise them for 1 to 2 years before returning them back to the exact same river in hopes of them spawning and migrating the same. Atlantic salmon make these large migrations all the way up to Greenland and back, and that's obviously a very far distance and potentially hard to navigate. So whether the salmon that have spent a year or 2 in captivity still have that instinctual behavior of knowing where to go and when is really important. And Atlantic salmon are really unique in the fact that they can repeat spawn. So their counterparts, the Pacific salmon, can't, but Atlantic salmon can reproduce multiple times, and it would be amazing to see if the supplemented salmon from our SAS program would be able to do that as well. Smolt-to-adult supplementation, it has been used before, but nothing's been ever tested to see whether these fish behave and migrate the same. That is really the whole point of this project, is to determine whether this SAS approach is effective at all for that. We're also using really cool technology.
The main type of tag that we're using is called a Vemco V13A tag, and the A stands for acceleration, and that means that we can track the really fine scale movements of the animals. And having that really fine scale activity transmitted with the tag allows us to see during which times of the year, in which parts of the river, during which times of the day are the animals most active.
These aren't exactly rare to use, but I think it's very important in this project because we can compare how exactly the wild versus the SAS salmon are using their energy, which is really important in how they're moving and surviving. We went up to Cape Breton Highlands National Park and deployed all of our receivers in strategic areas within the rivers. We then transported the tagged SAS salmon back into the river and hoped for the best. We then basically just sit and wait to see what the receivers pick up. But in the meantime, we need that wild salmon comparison. Unfortunately, the Clyburn Brook, where this project is stemming out of, has too few wild Atlantic salmon left to put into jeopardy of our tagging procedure. So we had to look to other local nearby rivers for our wild comparison.
We brought in the Sheddycamp River as part of the project to have some sort of comparator to better understand how the fish in that river that haven't undergone this Smolt-to-Adult supplementation, how different are these wild fish from these supplemented fish? Is there evidence of some sort of deficit that we need to focus on in order to improve or optimize the Smolt-to-Adult supplementation?
My name is René Aucoin. I am the president of the Sheddycamp River Salmon Association.
My name is Joanne Baker. I work with Renee. I'm the project manager for the Sheddycamp River Salmon Association.
Well, we started in the early 1980s, so fairly long time ago, and I was one of the co-founders. Initially, our interests were not necessarily in conservation. It was more on the fishing side, and I think this is more similar to many other organizations. Initially on the fishing side, we wanted to work with Parks Canada to try to improve access and other things, but over the years the organization started focusing on strictly conservation, salmon conservation. This particular river, the wild fish are still here. It hasn't been tampered with very much in the last 50 years. We have an excellent number. It's not a large number of salmon, but anywhere from 300 to 700 fish on a really good year that come into this river. And so it's a wild fishery, and it was a good fit, I think, to compare wild fish to fish that were raised in the Dal Aquatron.
We work with the park on a variety of different projects, on restoration projects, on habitat assessments, water quality monitoring projects. So we were reached out to by Sarah Penny, an ecologist with the park, asked if we wanted to collaborate on this project. And of course we said yes, and yeah, we were put in touch with some of the other researchers, and we were really happy to be involved.
We were asked to participate, number one, as anglers, and anything else that we could assist with. And for our organization, I think it was really interesting. This was very new in this particular setting, like to actually catch fish and operate on the fish.
Yeah, we got to help with both, like Renee said, recruiting anglers, but also deploying receivers. So we were basically asked, like, maybe which locations in the river would it make sense to have receivers deployed in. We got to help with that, also with the actual angling and the surgery as well. So that was, like Renee said, a first for us, but a really exciting project, and we're looking forward to doing more work like this in the future.
Yeah.
Because we were angling for kelts, so salmon that had already spawned, this was done in early December in this case. And normally during that time of the year, we're not doing a whole lot of work. We're not working in the river beyond the end of September. So we're typically more focused on reporting, applying for new funds for projects for the future years. So it wasn't taking away from our other activities, but I mean, for the anglers that we had involved, it was 4 days of angling, Renee?
4 or 5 days. We had 6 or 7 anglers involved. They were really keen. Once they saw the process, and particularly one important fact is we found out later that out of all the fish that had been operated on, all survived very well over winter. So we were pretty amazed at that, because we were iffy, what's going to happen to our fish,
Everyone loves the salmon, right? And we're concerned that even if one salmon may have been lost in that process, there were some initial concerns. But.
Yeah, we were watching them operate, the fish upside down, cutting the fish open. We had never seen that before, and what's going to happen to that fish? But they all survived. Wow, it was pretty amazing to hear that when Natalie told us that.
Relatively new to us working with academia, but we're excited by it, and we're hoping to do more.
I think over time things get elevated. So now we've been working more and more on science stuff, and because we've been around a long time, and I think we're hoping to still be around for a long time, and I think we are recognized as a small organization, but which has some capabilities. Jillian has a master's degree, and so very knowledgeable, and I think it's being recognized, throughout Eastern Canada that we're there, and if there are any universities or projects, if it's within our means to help, we'll do it.
Collaboration definitely played a major role in this project. We've been able to do so much work beyond our boundaries at Parks Canada, and we've been able to learn so much information that we couldn't have done without external researchers and these external projects that have contributed to us in the park, but we've also been able to contribute to them as well.
Ocean Tracking Network has loaned the receivers for this project, so all of the receivers that we're using to track the salmon moving within the Clyburn River, and all of the receivers that we're using at the same time to track the fish moving within the Cheticamp River, are all from OTN's loaner program. We use tags that will last for multiple years, and these fish that we've tagged will move out of these rivers. They'll move out of Clyburn, they'll move out of Cheticamp, and they'll go out to the ocean. And we expect that these fish will probably get detected on the Cabot Strait line or the Strait of Belle Isle line, and all those data will come back to the OTN database, and we'll be able to really effectively see where our tagged salmon have been detected in Atlantic Canada and potentially beyond. So we may be able to get a really broad story, a great sense of where the Cheticamp and the Clyburn salmon are migrating because we have access to this distributed infrastructure, this network of receivers with the data sharing and the ability to aggregate all that information through the database and efficiently know quickly where our fish have moved.
My favorite part of working on this project has definitely been the collaborative aspect. We've gotten to work with so many local groups and academics and other people who are just so passionate about Atlantic salmon. It's definitely a super dedicated group of people, and it's really awesome to be part of that movement and to get to see what these other groups are doing and to contribute as Parks Canada to do what we can for this endangered species.
OTN's really excited to support this project. Specifically in Nova Scotia, Atlantic salmon is a really high priority species, very, very highly valued and very economically important for the recreational fishery. And Nova Scotia has many different designatable units of Atlantic salmon, distinct populations. So just in this project, in Clyburn Brook, those salmon are part of the Eastern Cape Breton designatable unit, and in Cheticamp River, those salmon are part of the Southern Gulf of St. Lawrence designatable unit. Ocean Tracking Network has contributed to help tag these fish. So we had Cassandra Hardery from the field team up in Cheticamp helping to tag the salmon in December with Natalie. I helped to tag some of the salmon myself from Clyburn Brook in the Aquatron in September. And we've been really happy to be able to provide receivers, provide expertise, and to support the project however we can to better understand these two populations and to support the science on these two designatable units of Atlantic salmon.
I have met so many incredible people and learned so much from their experience. It's been amazing to be a part, again, from an academic point of view, and then also seeing how Parks Canada conducts their research and being a part of Ocean Tracking Network and meeting all of the local anglers. It's been truly a pleasure to get to know some of just the coolest people in conservation right now.
Behavior is critical to conservation. We want to be able to know, like I said, how are these animals actually interacting with the habitat? How are they using the areas that are available to them? And part of this project with the salmon that we're studying in Clyburn and Cheticamp is asking questions about spawning and the post-spawning of these animals. So the behavior during those periods is quite critical. We know that they have to conserve energy. We know that it's super important for salmon to be able to survive the winter in order to go back to the ocean and potentially spawn multiple times. And what are the behaviors that are underlying that really critical phase of their life?
But some preliminary results is that we've seen all of the salmon detected on our receivers, which is great news. So this means that the surgeries all went well and they were exploring the river. In the Clyburn River, we saw that some of the salmon have moved pretty far upstream, which is a great sign. Usually spawning locations where the salmon reproduce are further upstream and better habitat quality. So seeing quite a few of them move up is hopeful of them reproducing. From some preliminary genetic analyses as well that Parks Canada has received back, we've seen that at least one of the Smolt-to-Adult Supplemented salmon has reproduced, created offspring, which is great. It was only one juvenile that was found to be an offspring from these supplemented salmon, but this means we know that they can at least be capable of it.